Hi, my name's Stuart, and this is my project in exploring the bioactivation contributions of a burrowing frog community. Soil health is critical to agriculture as it faces increasing pressure from climate change, increasing the severity and frequency of extreme events such as your droughts. When considering soil health, animals such as your worms and ants tend to dominate not only the literature but the general public's knowledge, often leaving out other groups of animals such as your burrowing frogs. Now, burrowing frogs are found in every continent except Antarctica and spend most of their life underground, periodically emerging to the surface to breed and forage and then retreat back under. Now, this burrowing not only aerates the soil but improves water infiltration and root growth, while also mixing organic matter through the soil profile. However, due to the underground lifestyle, traditional techniques struggle to study burrowing frogs. However, novel techniques such as PAM or passive acoustic monitoring shows promise in addressing these issues. Therefore, this study aims to not only explore the contribution to soil movement from a burrowing frog community, but also is examining the viability of using PAM in monitoring these frogs. To explore their contribution, the site was visited weekly, where two 150-metre transects were conducted, spaced 5 metres apart just to ensure the same frog was encountered twice, while an acoustic recorder was also set up to record 24/7, capturing any frog calls. Frog density was calculated over the entire duration of the study period, assuming no recaptures, and soil movement was calculated using burrow sizes from existing values in the literature. In total, 3 species were found at the study site. Just before I go into those, this map on the left-hand side shows the distribution of each frog in the green. So first we have Cyclorana albigotata, as you can see here, mostly down your east coast. Next, we have Cyclorana brevipes. You can see very similar distribution along the east coast. And finally, Platyplex monatum, as you can see here, up along the northern coast, WA Northern Territory, all the way down your east coast down to New South Wales. Just before I start this graph, it is important to note, as I mentioned previously, these frogs are hard to study, so not all burrow depths were known or available in the literature, just so some data substitutions had to be made. So there was no data for Cyclorana brevipes, so I just had to use data from Cyclorana albigotata, and also no data for Platyplex monatum, where I had to use data from its close relative Neobotrachus aquilonius. So now that I've got that out of the way, this graph basically just shows a estimated soil moved over time between the 3 species, using the transect data, and just one major assumption here that all frogs we saw were individuals, so they've gone up to the surface and retreated back underneath. As you can see here, all burrowing, or the peak in burrowing, occurred between all 3 species in your mid to late December period, and then both your Cyclorana albigotata and Platyplex monatum both having significantly higher estimated soil moved when compared to your Cyclorana brevipes. And again, this pattern is repeated in this graph, which uses the estimated density that was calculated throughout the study period, and it again just shows that Cyclorana albigotata and Platyplex monatum both had significantly higher estimated soil movement when compared to Cyclorana brevipes, with Cyclorana albigotata topping out an estimated 300,000 cubic centimetres moved, whereas Platyplex monatum hovers around 200,000 cubic centimetres moved. Lastly, PAM was not able to identify individuals, therefore was not used in the calculation of soil movement. However, it was able to effectively identify calls of each species, therefore showing PAM could be a relatively cost-effective and passive tool used to monitor and detect the calls of these frogs. This study highlights the lack of understanding of these burrowing frogs and the benefits they provide. However, it also provides 4 key points for further research. The first one is quantifying the burrow depths of these frogs, and the second one is identifying the effect of soil type on burrow depths. As I was researching for this project, soil type was constantly cited as one of the main contributing factors to burrow depths. Third is increasing both the spatial and temporal scale, as the study was only over 6 months and a very localized zone. Fourth is identifying any other soil benefits burrowing frogs provide other than soil movement to farmers and agriculture.